Swayam Prabha. Digital India. Educated India.
Okay, so in this lecture I'll be talking about pectin. So pectin is also one of the biopolymers that can be obtained from fruits, vegetables, and so on. Compared to starch and cellulose, this pectin is a little bit more complicated to work with, but they have been already used as a gelling agent in confectionaries and fruit jams. And there is a lot of focus on using pectin as a edible film, a packaging film, and I want to cover certain aspects of pectin in this lecture, looking at sources of pectin and what are the structures, the molecular structure of pectin, and how do we extract pectin from sources, and then how do we form films with that, what are the innate properties of pectin and how we can reinforce. There's an upcoming area in pectin called thermoplastic pectin, like starch, for example, is now converted into thermoplastic starch, so you can extrude, you can process it thermally. Similarly, pectin can also be made thermoplastic. And also foams and aerogels is another upcoming area for pectin. So pectin is taking a center stage in this new kind of material for biodegradable or biocompatible food packaging films and packaging in general. Okay, so that's the context of today's lecture. Okay, just to kind of give you a heads up with what pectin is. So as I said, pectin is a biopolymer. That means it can be derived from biological origins, okay, from a renewable source. And it is a kind of a polysaccharide, but we call it as a heteropolysaccharide because it consists of 2 or more components, okay. And it's a major multifunctional component of the cell wall in many plants. Okay, so the pectin polymer is found in the cell walls of many plants, and it is actually, pectin is available. You can actually buy it on a commercial scale or industrial scale, and in these cases, pectin is extracted from cell plant walls using a lot of techniques, extraction techniques. I will briefly discuss about that at later stage. Predominantly, pectin is extracted from apple pomace or citrus peel. So that means once you have taken juice out of these fruits, apple and citrus fruits, the remaining biomass can be a rich source of pectin, and then you can do any sort of extraction mechanism to extract pectin. And why pectin is interesting? Well, it is not food, it is actually food derived. Okay, so it's basically food waste based material, so it's very low cost. Whereas if you consider a starch, corn starch or rice starch, these are food materials. Okay, so when we try to make polymers out of those food based material, then we are affecting the food security issues. Whereas pectin, on the other hand, can be obtained from food waste or fruit wastes. Okay, so therefore it is low cost and it's widely available, okay, from juice processing industries, and it's a natural polymer, so it is biodegradable, it is not toxic, it's compatible with biological systems, biocompatibility. So it kind of ticks all the things that we want for a polymer to be, okay, biodegradable, low cost, cheap. And as of now, it is extensively used in food industries with regard to fruit jams and confectionery items. But using pectin for commodity plastic is one potential that we can look into it. Okay, so it has a potential material for the formation of edible films, coating of fruits to improve the shelf life is other possible applications. Now, if you look at the structure, all polysaccharides are made up of sugars. But sugars also, there are different kinds of sugars. Okay, so depending upon how the sugar units are linked, what kind of bonds are used in this linking, the structure can be quite different. So broadly speaking, you can say pectin is like a copolymer. Okay, that means it would have 2 or 3 components linked together chemically, like AB, some kind of repeating units. And here we think of block A and block B. Okay, it broadly consists of two blocks, A and B. This block A is called this homogalacturonan. In short, we say HG, homogalacturonan. It is a linear homopolymer and it consists of galacturonic acid. The galacturonic acid is represented as a gal A. Okay, and so in the entire pectin, you have certain region which is made up of block A, right? And this block A is called this homogalacturonan. Okay, and this homogalacturonan block consists of this galacturonic acid, and this part of this chain is called smooth chain because it is linear and nicely packed. In any given sample, pectin sample, typically you will have between 50 to 70% of the entire polymer is made up of this block A galacturonan, homogalacturonan component. Now, if you look at the chemical details, you can see that this is the bond that links two sugar molecules. Okay, and this we call as a 1,4 glycosidic bond. Okay, the oxygen bond that links the two sugar molecules, we call them as glycosidic bond, and it's called 1,4 because if I look at this particular sugar molecule and if I start numbering 1, the carbon here 1, this is carbon 2, this is carbon 3, this is carbon 4, this is carbon 5, and this is carbon 6. Okay, similarly if I take this unit, I would call this is 1, this is 2, 3, 4, 5 and 6. Okay, so if you look at these two molecules, you can see the bond is between the first carbon of the one sugar unit and fourth carbon of the other unit. Okay, so you can see that all this oxygen groups here are nicely lined up. That's what makes them it's a linear polymer and quite homogeneous. Okay, so the sugar molecules are linked via this 1,4 glycosidic bond, giving rise to this kind of linear structure, right? And the other component, which is called the block B, is called rhamnogalacturonan 1, RG1 in short. Okay, this rhamnogalacturonan is actually a heteropolymer and it consists of repeating units of the gal A and rhamnose disaccharide backbone, and we call this as a hairy region because this is very amorphous. And any given pectin sample can have between 20 to 35% of this rhamnogalacturonan RG1. And then if you compare the structure of galacturonan and rhamnogalacturonan, you can already see this, the bond formation is quite different now, because you see here the oxygen is in the top, in the next molecule it comes down, then goes up and then it goes down. So it kind of in a different arrangement. Okay, and this is because the bond that connects two sugar units are 1 and 2 glycosidic bond. That means if you look at this, this is the first carbon, this is second carbon, third, fourth, fifth and sixth. And if I take this one, here you have this is 1, this is 2, this is 3, this is 4, this is 5, right? So in this case what happens is the 6 is this one. Yeah, so you can see the bond here is between the first carbon of one sugar unit and the second carbon of the other sugar unit, and because of this linkage you see the sugar molecules gets twisted, and then you have an alternative arrangement of up down kind of structure. So in the homogalacturonan you see that all of these chains are arranged in one particular way. Okay, so if I have to show where this oxygen group is present, if I show some sort of direction to that, you see that all this, if I stretch this chain, you will see all this oxygen is kind of pointing in one direction. Whereas in the rhamnogalacturonan, you will have alternative arrangements of up and down kind of a arrangement, just because the bond links, the bond that links the two sugar molecules are different in the homogalacturonan and in the rhamnogalacturonan. Okay, and not only these two, there are also other components, something called rhamnogalacturonan type 2, which may consist of about 10% of the total chain, and then there is another component called xylogalacturonan, or XG in short. In some sample you can have raminogalacturonan 2 and xylogalacturonan also present in this sample. This is what makes pectin quite complex molecule to work with. Depending upon where you extract pectin from and which method you are using to extract, you may have a compositional variation between homogalacturonan, raminogalacturonan 1 and 2, and then xylogalacturonan. Okay, and that's what makes it a little bit difficult to study and reproduce the pectin-based material. I think I have a kind of a geometric model of this pectin. So what you can see here is a schematic of how different polymers subgroups are linked together. So you can see that the majority of them are this galacturonic acid, shown as this diamond like shape. You can see that this entire region, you can see a linear arrangement of homogalacturonan, and then the raminogalacturonan, you can see that it forms this kind of a branching units, right? And this pink one that you see here, that is a ramino sugar. Okay, and then in a real sample you can have so many other sugar molecules also, xylogalacturonans. There is a section where there is a xylogalacturonan. There is raminogalacturonan part 2 also, where you can see how many different components are there in these two, and this is what makes them little amorphous and hairy. Okay, whereas the homogalacturonan is pretty, linear and quite smooth. So based on the type of source and the extraction method, the composition of this pectin can be quite variable. Okay, so I'm not discussing all the other possible sugars that is there, but it's just a kind of a schematic. Now pectin, when you try to work with pectin, you have to look at what kind of pectins you are working on, and despite that there is a lot of variability in the composition, there are broad classification called low methoxy and high methoxy pectin. These are based on something called degree of methylation. You see, if I go back here, I have made a mess of this figure, but you can see in all this sugar unit you have the COH groups. Okay, the galacturonic acid has the COH group, and that acidic group, carboxylic group, can be esterified with the methanol. Okay, that's what called methylation. So you can see that here you have unmethylated carboxylic acid. This group is actually methylated. You can see there is a CH3 groups coming in. Okay, so depending upon the overall, in the total number of galacturonic acid, how many acids are esterified with methanol, that is what defines the degree of methylation. Okay, so the carboxylic group of galacturonic acid is esterified with methanol, giving you this kind of group. Okay, so this is unmethylated, unesterified, this is esterified. Now if you have 10 such groups, of that how many groups are esterified with methanol, that's what tell you degree of methylation. If you make all the acidic groups with esterification, then you have 100% degree of methylation. Okay, when you have none of the carboxylic acid groups methylated, you have 0 degree of methylation. So broadly speaking, you see something called high methoxy pectin, or HMP, or low methoxy pectin called LMP. This is depending upon whether the degree of methylation is above 50% or below 50%. Okay, now depending upon to what extent this is esterified, the physical properties can be quite different. For example, in the case of high methoxyl pectin, it is capable of forming gels in the pH range 2 to 3.5 with a high concentration of additional sugars or plasticizers like sorbitols between 55 to 70%. Okay, so that means you can make a gel with high methoxyl pectin also with a pH range in the acidic range, and then with an addition of some additional sugar molecules at a very high percentage. Whereas if you consider the low methoxy group, which contains less than 50% of esters, they can form gels over a wide range of pH from 2 to 6 without adding any further sugar. Okay, so the gelling property of pectin lot depends on the degree of methylation. Okay, if you want to make a good gel, low methoxy is easy to make. In high methoxy also you can do, but provided that you change the pH in a narrow range, and then you add additional sugar and sorbitol, right. There is one more thing, this is called methylation of pectin. There is also something called acetylation also, acetylation of pectin. In that case, what happens is that this one of this OH group that you have here, this combines with acetic acid, and then you will get basically this group will become O COO and CH3. Okay, so this becomes one of the group become acetylated. Okay, while the carboxylic group can be methylated, the OH group here can be acetylated also. So depending upon the degree of acetylation, the property of the pectin also will change. So it is subjected for many kinds of further chemical modifications, especially with methanol and ethanol and other things. Alright, so now having understood it is a very complicated or complex blocker polymer, majorly containing linear homogalacturonan, and then having about 20 to 35% of rhamnogalacturonan, maybe in addition xylogalacturonan and etc. You have different degrees of methylation, and hence the properties can be changed. Now, if you look at how pectin is extracted, the extraction technique and the source, whether it is apple or citrus, based on the source and based on the extraction, the quality and the yield of pectin can be quite different. So quality, we mean what is the percentage of homogalacturonan and rhamnogalacturonan and others, and yield means for every 1 gram of raw material I have, how much pectin I can extract. Okay, so both of them depend on the source and the extraction technique. Typically in this extraction, very harsh acids are used because you want to go to very high acid concentrations or very low pH, where you can extract pectin from other materials. Typically HCl, lactic acid, nitric acid, these are used, and they are known to give high yields of pectin by removing the bond between the pectin and the cell wall matrix. Okay, in some cases alkali based extraction is also possible. Sodium hydroxide or even enzymes can be used to extract pectin. There is always a need for looking for a greener, environmentally friendly and sustainable acids for extraction of pectin. And some of the green organic acid that are being currently researched on include citric acid, acetic acid, malic acid, tannic acid, and so on. Okay, so these acids can be produced from bio origin, and these acids can be used for extracting pectin. Okay, as an alternative for harsh acids. Again, we are talking about sustainability. It is not just about the product that we make, but also the process with which we make the product is also important. Okay, so if there is a way we can eliminate these harsh acids and bases and then work with simple green organic acids, that is one area in which a lot of research can be diverted to. So here I have given one example of the process flow for acid hydrolysis. So typically what happens is, if you want to, let's say, use apple pomace for extraction, so after apple is crushed and then juice is obtained, the waste pomace is dried as a powder. Okay, and the dry pomace is ground into a flour. You want to convert into a flour because when the particle size is very small, the extraction can be readily obtained. So there is a lesser mass transfer resistance and so on. Okay, so they can have basically high surface area particles. Then what happens is this dry powder is treated with acid, and typically the pH goes below 3 and the temperature can be up to 60 to 100 degrees Celsius, and constantly you stir over half an hour to 6 hours. Okay, these are the range of pH values, times and temperatures. It changes depending upon what is the pH you use, what is the source we use, and what kind of agitation or mixing we do. Okay, once you do that, the pectin will precipitate in this system. You can add a cross-linking agent or you can use a non-solvent to actually precipitate, filter it and then wash it, and then you have your pectin. So it's schematically shown here. You start with your apple pomace, which is basically the biomass left after you crush the fruits to make juice, and then you dry into a powder, and then you can see that there is a series of grinding process from a coarse to a very fine powder, and then you add your acid less than pH 3 over a range of temperatures, boiling temperature, and over half an hour to 6 hours, and then you basically get the pectin thing, and then you can wash it and then cast into various films. So that's a typical method of extracting pectin from acid. So here the acid can be HCl or nitric acid, other acids. So here is a comparison between various kinds of extraction. So if you look at acid extraction that I shared in the previous slide, so pH is between 1.5 to 3, temperature can be of the order of 100, the time is about 1 to 3 hours. Acidic extraction is the one that gives the highest yield of pectin and most preferred also, and it can actually extract the high methoxylated pectin. You can see that the molecular weight can go from 10 power 4 to 10 power 5 daltons, and in this particular acidic extraction, you predominantly get the homogalacturonic acid, which is a linear smooth region, while you destroy most of the amorphous or rhamnogalacturonic acid. So when you try to process apple pomace with acidic extraction, you mostly dissolve or destroy the hairy amorphous region, and you are mostly going to get the smooth region. Okay, so here is an example that the yield can be roughly between 60 to 80% of degree of esterification is possible in this case. If you look at alkali, in the alkali you can see the pH is in the alkali pH 9 to 13 using sodium hydroxide or any other alkali, and temperature can be anywhere from room temperature to 80%, 80 degrees. These ranges are given based on what is the previous work has been done, and it is shown that usually the alkali extraction gives a very low yield of pectin, and that is because it produces the low methoxylated pectin, and it extracts mostly the hairy or amorphous rhamnogalacturonin, while it destroys the HGA, the homogalacturonic acid. So you can see a stark contrast between the acidic and alkali extraction. If you want to get the linear part of the polymer, the homogalacturonin, you would do acidic. If you want to preserve the amorphous or a hairy region of the polymer, you would do alkali extraction, and it kind of gives you a lower degree of esterification than acidic esterification. The third approach is this enzymatic extraction. So here the pH range is between 3 to 4.5. Temperature is a little lesser than the acidic or basic extraction, but the time you can see it can go up to 4 hours to 24 hours, because we know enzyme reactions are quite slow than the acidic and alkali extraction, so time is quite large here. However, enzymes are actually very selective, and they do not disturb the entire structure. They try to preserve the actual pectin structure intact, and you can see that from the apple pomace, the enzymatic extraction can yield up to 97% high degree of methylated pectins. The examples of enzymes that are used are esterases, hydrolysis, and lysase. Okay, these are the enzymes that are used. It also means that if you use a pectin-based film, these enzymes can actually degrade after the product is actually littered or thrown away. Okay, so as one would expect, enzymatic extraction is going to be quite expensive, because you need enzymes, you need active enzymes, so purification of enzyme and is quite costly, and doing it in large scale is also a challenge. However, the advantage is that you could have much lesser degradation of pectin from the native state. Okay, we saw that in the acidic extraction, it preserves the linear part of the polymer while destroying the amorphous part, whereas in the alkali extraction, it destroys the linear part but preserves the amorphous part, whereas enzymatic extraction would actually preserve the entire structure, both linear and amorphous together, but time is more, cost is more. Okay, so that is but it's a eco-friendly approach, as I said. So this is a kind of little comparison between various kinds of extraction that is possible. Now, there are a lot of literature on pectin-based films, especially for food application or food packaging applications, and a method called solvent casting is used for this case. In this case, what you will do is you will start with your pectin powder, okay, which is extracted from one of those previous extraction methods, and you would dissolve it in a solution, typically water or acetic acid. You would change the pH to make the right consistency, and then you will cast into a casting machine. You will let the solvent evaporate and allows the formation of film, and then you can peel off the film. Okay, so one more thing I should tell is that the substrate is also important. Typically, the substrate is held as a hydrophobic substrate, so that once the film dries, it delaminates easily and you can peel it off. Okay, if you have a hydrophobic substrate here, it may get stuck on the surface, so it may be difficult to peel off. So you can optimize the substrate chemistry, and you can also control the film thickness by changing the polymer concentration. You can make a thicker film, thinner film, all that. And if you want to add any additives, that is also possible. You can add an antimicrobial agent. You can add some more functional additives while the film forms. Now, if you consider just the neat pectin, okay, the virgin pectin alone, you will see that these pectin films are actually quite hydrophilic, and they are quite brittle. Okay, they are easy to tear if you are employing it alone. Therefore, they are not actually suitable for a packaging application per se. Okay, you can see that if you leave pectin film on the table, after some time you will actually absorb a lot of moisture, become very sticky. Okay, so that is one issue with native pectin. So especially when the solvent evaporates, you will see some sort of a cracks and curling. The film becomes a little unstable, and it has a very high water permeability, and it has a very low strength. Okay, these are all problem with the native pectin. Therefore, if you want to use pectin for food application or food packaging application, you have to modify pectin with certain additives. Okay, for example, usually it is a cross-linking agent is added, so that the chains in the pectin film can actually interlock or cross-link and give a better mechanical property. Sometimes you would use plasticizers. We saw use of plasticizer in starch and other materials, which will make the chains a little bit more flexible, gives more elongation, avoids the brittleness. Okay, so these are typically added, and in addition, you can also add antioxidants and nutrients, antimicrobials, sometimes enzymes, depending upon what functional role you want the film to act upon. You can actually, if you want to have a for example, let's say you want to pack fruits, okay, and then you want to look at the ripening stage of fruit or any sort of fungal infection, you can put a marker, and then the film itself can actually integrate these markers, and the colorimetric sensing, color change can be detected to see. Okay, so and so here I have given some example based on the literature how pectin films are formed. So typically about 2 to 5% pectin is used for making films. You will also see sometimes pectin is combined with glycerol. Glycerol acts as a plasticizer to make the film a little bit more flexible upon drying. So in another recipe, the low methoxyl pectin is mixed with the glycerol and the calcium chloride. So here calcium chloride is added because it cross-links the polymer chains, gives better mechanical strength. In another recipe, LMP, glycerol, that is plasticizer, cross-linking agent, and also some peptide from pepsin hydrolysis is also added to give additional mechanical property. Or you can also mix with other polymers, like pullulon is another polysaccharide. You can mix pectin and pullulon in 50-50 ratio and then cast into film. Since pectin is from a bio origin, it blends nicely with other hydrophilic biopolymers like pullulon, starch, and so on. And another example, pectin, agar, zinc sulfide nanopetals. You can actually add nanoparticles also into the film to reinforce. Okay, so these are not very exhaustive list. This is just a representative list. If you go and search for literature, you will see a whole lot of recipe available for casting pectin based films. So now I was telling that pectin is a hydrophilic polymer. You can make a solution out of it and then you cast a film on a surface and let the film dry of solvents and then you can peel off. But making a thermoplastic version of pectin is also an upcoming area. There are only few papers on this topic, but it is something that you can think about if you want to work on pectin. This is one area that you can look at it. Just like how thermoplastic starch is being made and used for film formation through extrusion process or blow film process, pectin can also be used in the similar way. There are few papers. For example, in one of the papers I found that the high methoxylated pectin is mixed with the glycerol and the citric acid in certain proportions. So 49 weight percent of HMP, 30% of glycerol and 21% of citric acid. The reason to add citric acid in this particular process is that they say when you do when you increase the temperature to let's say 90, 100 degrees Celsius, the presence of citric acid actually prevent oxidation of the pectin and gives the thermal stability. Okay, so so they found this particular ratio seems to be the right one. So they used a co-rotating twin screw extruder with a length to diameter ratio of 40 and a screw diameter of about 20 mm and with the 7 heating zones. Okay, so that's a long 40 aspect ratio twin screw extruder, and this particular composition is put there and the temperature profile is shown here. So the feed, it's almost like a room temperature 33 degrees Celsius, and the temperature gradually increases up to 100 and towards the nozzle it's about 90. You can see that there are 7 different temperature settings ranging from 35 to 100 degrees Celsius and a screw speed of about 100 RPM is set. So with this process they were able to extrude this into a pellet. Okay, and then this pellet is then thermocompressed using a hydraulic press to form a film, to form a sheet. And they have also looked at different conditions. 15 grams of pellets is pressed at 110 degrees Celsius at the following conditions: 110 of weight expressed for 3 seconds, or 210 for 2 minutes, or 310 for, these are different operating conditions, and they were able to get a film of 15 by 15 centimeter. And once the films were formed and they were actually cooled at a room temperature. Okay, so this is one example of how to make thermoplastic pectin and how you can use compression molding for making films. So here is an example of those films. The one you see on these two are actually pectin films that are formed at a pH 2 and pH 7 using solution casting, the usual approach. Okay, so you take a pectin and at certain concentration and regulate the pH to 2, and then you cast into a film, dry the film, and then you cut into a shape. And this is same as that, but the pH here is 7. Now they have done 2 more experiments which is based on extrusion. So they make this thermoplastic pectin and then they use this hydraulic press to form a film. These 2 are actually made with the 2 different pH of the original original solution. Okay, and you can see there is a small color change. This color change is because of can you explain why this color change happens? Any example, any idea? What is pectin made of? Sugar. So what happens if you heat sugar? Caramelize. Yes, that's exactly what happens here. Because you are heating at 100 and 110, some of the sugar molecule actually caramelizes, and that's what you see here as a color change, right? Whereas the one at the first 2 sample, they are casted at maybe 60 degrees or 50 degrees, and then the solvent evaporation happens. So you can see there's not much of a caramelization happens here. But in the last 2 examples, the caramelization leads to this kind of color change. All right. So as I said, this is one potential area one can look into it. There is another upcoming area that is based on foams, pectin foams and pectin aerogels. You may recall that we were talking about cellulose foams and cellulose based packaging, foam packaging. So in the pectin foams, the idea is you know, ability to make a very porous structure that could be used as a foam for packaging or insulating material and other things. Okay, this is also an upcoming area. Not much work has been done yet, so this is also something for you to look after. So I have given one particular example. More detail can be found in this paper. So here what they have done is they have taken pectin powder and in water at 60 degrees Celsius, and then they have kind of stirred at 400 rpm over 4 to 5 hours for complete dissolution of pectin. After complete dissolution, the solution pH is adjusted to in acidic condition 1 to 3, and then the entire solution is poured into a mold, okay, silicon mold. And if you want, one can also add calcium chloride as a cross-linking agent to induce gelation. Okay. Now this entire solution on a mold is kept at room temperature over 2 days. Okay, you can see it's quite a long time. Okay, so if you can come up with a better processing condition which can form a gel and take the shape very immediately, then you have something to work on. Now you at 48 hours the gelation is complete and the gel would have taken the form given by the mold itself. Okay, so now if you look at the gel, it has got pectin, it has got let's say calcium chloride, it has got water, lot of water. Okay, so now you need to remove water. One approach is to slowly evaporate this water. If you do that, you may affect the structure of the the porous structure of the film. So what they did on the other hand is they slowly exchanged water with a solvent like ethanol. Okay, by treating this mold, the product in dipping it in ethanol water solution, they were able to exchange water with ethanol. Once you have ethanol filled gel or called ethanol gel, then they do something called supercritical CO2 extraction to dry the ethanol, okay, without affecting the porous structure. So as you can see here, these are some images that shows how porous this networks are. The thread like, the fiber like structure that you see, that is basically, you will see if you open let's say if you open orange, and if you look at the inner wall, you will see this kind of very stranded structures. Okay, so they're able to create a very similar kind of a porous structure by doing this process. Okay, so and the application they did for this is to get thermal conductivity and for insulating purpose. But then this method is very useful to make a foam, if you like, okay, foam or a cushion like product that can be used for packaging application. Okay, this is also an upcoming area. A lot of research can be done on this also. Okay, there is one more research that is possible, is to do what is called pectin biosynthesis. Right now what we are looking at is, we are going to apple pomace or citrus fruit pomace, take the fruit waste and then do alkali hydrolysis, acid hydrolysis, enzymatic hydrolysis, and extract the protein the best possible way, and then process it for film formation, thermoplastic pectin, all that. Another possibility is to recreate how cells or how biological systems produce pectin, and a lot of work has been done in identifying the right enzymes that actually are instrumental in converting sugar into a polymer, sugar into a pectin. It is said that pectin is actually synthesized in the Golgi apparatus by an enzyme called Golgi localized glycosyl transferases. Okay, so this glycosyl transferases is an enzyme, and what it does is it actually takes glycosyl residue, which is basically a sugar moiety, from the nucleotide sugar, and it takes this sugar from nucleotide sugar and then attaches to this pectin, growing pectin chain into oligosaccharides and polysaccharides. Sometimes the pectin residue is also modified by methyl transferase. This is another catalyst or enzyme that will actually make some of this into an ester group, right? And or you can also do something another called this acetyl transferase. There are 3 enzymes that are possible that are involved in the formation of pectin in biosynthesis pathway. So one way to look at this is, can you come up with the programmed enzyme combinations that will take sugar and convert the sugar into oligomers and polymers in a controlled conditions, so that you can precisely control the type of sugar. If you want to build only the homogalactoronine, you can do that, or if you want to change the degree of esterarization, you can change the amount of the methyl ester transferase. If you want to acetylate the polymer, you can also control the amount of acetyl transferase reactions. Okay, so this seems to be one area, again a lot of possibilities awaiting. So this is my last slide. Why I'm talking about pectin is because we are working with pectin in our lab. This is a paper that we published in last year in IGNOU. So here we just start with the pectin, right, and then we try to kind of improve the mechanical properties of pectin by adding needle shaped or rod shaped nanoparticles, iron oxide or hematite nanoparticles. You can see this is the native film, which is transparent, and then if you load with this nano ellipsoids, you can see the color, the rusty color, because this hematite is basically has this kind of a rusty color. And then what we do is, compared to the native pectin, which is cross-linked with calcium, so here we have put this hematite nanoparticles to be able to integrate nicely with the polymer. So with that, we could actually change or increase the tensile strength of the native pectin, and we can also increase or decrease, on the other hand, the water permeability that is required for packaging application. And then if you look at the native pectin film, the water contact angle, that is, we measure how hydrophilic it is by measuring what is called this contact angle. You put a small amount of droplet of water. If the water spreads, it would have a very small contact angle. If the water sits like a droplet, like a water droplet sitting on a lotus leaf, it would have very high contact angle. So we can see that by increasing amount of hematite nanoparticle, we are able to increase this contact angle from about 50 to close to 85 or so. Okay, that means water will not really go into the film; it will roll off pretty much. So if you want to know more about this particular work, you can you can look at this particular paper. There is a potential, as I said, pectin. We see that a lot of food waste based sources are available, which will not affect the food security. Okay, so there is a way in which we can source many food waste material and then extract pectin from those food waste material. There is a lot of opportunity to come up with a new processing technique which uses milder, greener, eco-friendly chemicals or acids. Okay, that's one domain one can work on. Then the second thing is, you can actually come up with a different kind of reinforcement for the pectin film using nanoparticles and additives and plasticizers, etc., to come up with a film that can be flexible, having high tensile strength, that would have very low water permeability. Okay, so that it can actually protect the food material or whatever that we are packing in. And the upcoming areas are thermoplastic pectin. How can I come up with a pectin formulation that can be extruded into pellet and then it can be compression molded, or it can be injection molded. Then about foams, pectin-based foams for packaging application or cushioning application is also another possibility. And thirdly, how can we synthetically make pectin using enzymes is also another area. Okay, so these are the areas that you can look for if you are interested in working pectin. Okay, so with that I'll stop here.